IB Bio students. All right, so today we're going to continue with higher level topic 10.2. Oops, and I just lost it. Let's go back. And we're going to be looking at polygenic traits, which is this last core content within this section. So we're going to be looking at polygenic traits. And let's start with the essential understanding. So here you have two essential understandings. One is that variation can be discrete or continuous. And two, that the phenotypes of polygenic characteristics tend to show a continuous variation. So first, let's differentiate how variation in phenotypes happen. So a particular characteristic, right, or a particular trait, for example, eye color, hair color, height, or in Mendel's case, pod shape, seed color, can either be discrete, discontinuous, or continuous. So when we think of monogenic traits, these are traits that are characterized by a single gene loci. And the gene loci is the position of the gene with the allele variations. So a single gene loci is a monogenic trait. And if you are a monogenic trait, like in Mendel's peas experiments, so I'll write this here, Mendel's peas experiments, he looked at monogenic traits and they tend to exhibit discrete variation. So that means that you either express one of the phenotypes or the other. So in his peas, he either had green or yellow pea pods, right? Or the pea pod was either wrinkled or smooth, or the plant was tall or short. So those are examples of monogenic traits. Now, polygenic traits are the other side of the coin that we didn't really get into a lot back in 9th grade or SL Bio. And these are characteristics that are controlled by more than two gene loci. So again, the gene loci is the position of that gene and how that gene incorporates its two variations or its two alleles. So when there are more than two gene loci, the characteristic tends to exhibit what we know as continuous variation. And continuous variation is what we refer to as polygenic inheritance. So that means that the phenotype of the individual is going to lie somewhere along a continuous spectrum of potential phenotypes. And that continuous spectrum is what we describe as a possible bell-shaped normal distribution curve, or a Gaussian bell-shaped normal distribution curve. So let's look and see at how that is done. So as you increase, so in polygenic inheritance, polygenic traits, as you increase the number of loci responsible for a particular trait, then you also increase the number of potential possible phenotypes. So here we see how we go from a monogenic trait. So here in this gray illustration, you see a monogenic trait. And in this particular monogenic trait, you have just one gene loci, or just one position for that gene loci, that's contributing to a single trait. And remember that every gene has two variations or two alleles. So in this particular case, you see the gene loci could potentially express something along the lines of a heterozygous phenotype, where you see the dominant and the recessive alleles at the same time. Right? So this is what you studied back in the 9th grade with Mendelian genetics. You studied the law of dominance. So in this particular case, you would have a heterozygous individual. And if the law of dominance applied, not looking at the exceptions like the law of incomplete dominance, you would say that in this particular case, right, this individual will be more along the dominant side. Okay. But either variation could apply, right? Either the recessive, if you have a homozygous recessive individual, or a darker trait if you have a homozygous dominant individual. Now, as you move along through polygenic inheritance, notice that the number of gene loci contributing to a single trait increases. So now we've increased the number of gene loci to 2. So you could potentially have like a really light yellow or a medium yellow or a darker yellow, depending on how the alleles recombine, right? So it's not as clear cut as monogenic, where you either had the recessive homozygous, the dominant homozygous, or perhaps a combination of the heterozygous condition. Here you're more in a bell-shaped distribution of different possible phenotypes. Notice that as you increase the number of gene loci, so now you have 3 here, and if you start mixing those dominant and recessive traits, right, or those dominant or recessive alleles in different amounts, you can end up with an even greater phenotypic possibility, right? So you can have light pink or a little bit darker pink or even more darker pink or a dark pink. And notice that it's true as you move along this distribution, right? So when you incorporate 4 different gene loci, then you have more possibilities of a phenotypic distribution. As you add 5 gene loci, you have more possibilities here of a phenotypic distribution. And when you think of eye color, for example, and this probably will finally explain why people all have different eye colors, is because in the eye color, you have more than 1 possible gene loci. You can have up to 6 different gene loci. So that's why you end up having people with blue eyes or really light blue eyes or a darker blue, more of green eyes, and then more of hazel eyes and more of brown eyes. So again, you have a different spread here of the different possible phenotypic variations. Okay, so this is what we refer to as polygenic inheritance. More than 2 gene loci for a particular characteristic, like eye color, for example. Okay, so when we look at corn grain color, we see that it's controlled by 3 gene loci. So maize is corn. And notice that the grain color of corn can vary from white to dark red, depending on the amount of pigment that is being expressed. So notice here in this picture how corn expresses a form of polygenic inheritance or continuous variation. So here you see the 3 gene loci, and notice that the 3 gene loci could either have a homozygous allele combination, it could be heterozygous, or it could be homozygous dominant, right? So homozygous recessive, homozygous dominant, or even heterozygous allele combination. So if you cross a pure white line of corn with a pure red line of corn, you're going to get a hybrid, right? You're going to get somewhere in between. So you're going to get a heterozygous condition of the 3 gene loci with both the dominant and the recessive allele. Okay. So here in this bell-shaped curve expressing polygenic inheritance, you see how as you increase, right, the number of pigments, and in this particular case you have, not the number of pigments, sorry, but the gene loci, you increase the different possibilities of what the offspring could look like. So this is what we see in corn. in the corn grain color. You can have different variations of the color depending on what genotype you have. So notice that there's no real dominance in this particular case, right? It's just a continuous variation form. So in the activity that you are going to be working on today, you're going to be looking at all the different forms of continuous variations in corn as well.